Hi everyone, welcome to my YouTube channel. If you are new here, subscribe to be notified as soon as I upload a new past paper video with simplified explanation. Don't forget to like and comment. I will be more than happy to solve any doubts. Today we will be discussing Cambridge IGCSE 0610 Paper 6 Variant 2 from the October-November session, which is of year 2025. So, to begin with the paper, let's start with reading the question first. So here we read that a student investigated the rate of respiration in yeast using different concentrations of glucose. As I have mentioned earlier in my videos, that the first point that we need to do over here is to identify the independent and the dependent variable, which will always be present in the first line of this experiment question. So, to identify the independent variable, we are going to look at the rate of respiration in yeast with different concentrations of glucose. Now, what can we change in this experiment? We can change the concentrations of the glucose and we can observe or count the rate of respiration in yeast. So, this is how the changes sometimes occur in the paper. So every time we consider that the independent variable will come first in the sentence, but here the rate of respiration is the dependent variable and the concentration of glucose is the independent variable. Now, I will just highlight this. The rate of respiration is the dependent and the different concentrations of glucose is the independent variable. As we remember that the yeast is doing the work which is called as the anaerobic respiration. Anaerobic respiration means that it happens without or in absence of oxygen. Okay, so in absence of oxygen, less amount of energy is released. Yeast using this part of anaerobic respiration results in the process of fermentation, which will result in production of carbon dioxide and alcohol. A similar kind of activity will happen in the human body as well when the person is undergoing an aerobic activity. The levels of lactic acid when it increases in the body and there is less oxygen as compared to normal, that is the time a product called lactic acid will be formed in the human body. To continue with this question ahead, they used 5% glucose solution to make 1% glucose solution and 4% glucose solution. So they have started with an initial 5% glucose and they are making two different concentrations, which is our independent variable, that is 1% glucose and 4% glucose solution. The student used the method as follows. They took they labeled two small test tubes and labeled it as 1% and 4%. Using a syringe to put 1 cm cube of 5% glucose solution. So suppose here in the test tube I have 5 cm cube. They have taken another test tube where they had labeled 1% and they have added 1 cm cube from here to here. Okay, so now they have 1 cm cube, that's the volume of glucose solution that is added in the test tube which was labeled 1%. Put 4 cm cube of 5% glucose solution in the test tube labeled 4%. So the remaining 4 cm cube they have added in the test tube which they had labeled as 4%. Use a clean syringe to put 4 cm cube of distilled water in the test tube labeled 1%. So now they are adding 4 cm cube of water which will add up to the total 1 plus 4 is 5 cm cube altogether. Put 1 cm cube of distilled water in the test tube labeled 4%. So here they are adding 1 cm cube. Again 4 plus 1 will add up to 5 cm cube. So that is like a control variable that they are having the same volume of the solution in the test tube. 1% as I understand will be more diluted, or it will have more water potential as compared to the 4%. 4% is concentrated than the 1 cm cube solution. Pour the yeast suspension in the test tube labeled 1% until it is completely full. So the remaining part they have filled with the yeast. Keep the test tube upright and carefully place a large test tube over that. So you can see that this was our original test tube. On that they have inverted another test tube like this, okay, which is they have told mentioned it as large test tube, and now that large test tube they have inserted in a water bath containing warm water. Put the large test tube and the small test tube labeled 1% in a warm water bath. Start the stop clock and wait for 5 minutes. After 5 minutes, stop the stop clock and use the ruler to measure the height of the yeast suspension in the large test tube. So, the yeast has traveled from here using the glucose and water, it has moved out. So, the height of the yeast suspension in the large test tube after 5 minutes is what we are going to record using this particular measurements. Remove the test tube from the warm water bath and discard them. Pour the yeast suspension in a small test tube 4% until it is completely full. Repeat the steps 4 to 8 with the small test tube labeled 4%. So they are going to repeat the same thing again with 4%, and then they are going to measure the height of yeast suspension in the large test tube again after 5 minutes. You can see the difference on the screen. Figure 1.3 is a diagram of the water bath in tests in steps 8 and 11. This is the labeling for a 1% glucose solution and 4% glucose solution, and you can definitely see the difference in the height of the yeast suspension. Measure the height of the yeast suspension in each of the large test tubes shown and prepare a table for the results and record the measurements from figure 1.3. So, as we can see over here, that they have clearly mentioned that you have to record the results of the measurement in the table. So for the table, I have made two columns, okay, and it has a header line with the concentration of glucose solution which we measured in percentage, and the height of the yeast suspension in millimeters. And now I have put the labeling as 1 and 4 and 14 and 24. Okay, so that's when you measure it on the paper, you will get a similar kind of a reading. Since the percentage was put over here, I have put this specifically to highlight that since the percentage sign has been put over here, we do not put another percentage sign over here. Okay, so we have 1 and 4 and 14 and 24. That is what is just to be put in the paper. So how are you going to be marked on this question? The table drawn with minimum 2 columns and a header line. So that's what we have done. Appropriate columns and row headings with the units of for height. So we have put as millimeters. Recording two final heights, that's your 14 and 24. Okay, and the method recorded again. So that's how we are going to talk about 1% and 4%. Okay, so that's how the marking is going for this question. We move ahead in the paper. State the conclusion for the results of this investigation. Now be very clear when they use the word conclusion. Conclusion is not an observation. Okay, if you had to write an observation, you would have just said that as the concentration of the glucose increases, the height of the suspension is increasing from 14 to 24. But this is called as an observation. Conclusion means what actually occurred or why it occurred. So here, the higher the concentration of glucose in the solution, the greater is the rate of respiration of yeast. Okay, so that is going to be the conclusion, because that was the aim of the experiment. If I go back to the first page, they are asking you the rate of respiration in yeast with the different concentrations. So most of the time your conclusion will be on basis of the aim. So here I again repeat that the higher the concentration of glucose in the solution, the greater is the rate of respiration of yeast. State the independent and the dependent variables in this investigation. So the independent variable will be definitely the concentration of glucose, and the dependent variable is what we have measured with the scale. So that is the height of yeast in the large test tube. Describe how the temperature of the water bath could be maintained. So, thermostatically controlled water bath can be used to maintain the temperature. Explain why a clean syringe is used in step 3. So, if I go back to step 3, they said, before step 3, let's read what is happening. So, they said use a syringe to put 1 cm cube of 5% glucose solution in the test tube labeled 1%. And put 4 cm cube of yeast 5% glucose solution in the small test tube labeled 4%. Now is the step that we are looking for, which is saying use a clean syringe to put 4 cm cube of distilled water in the small test tube. Now, if the syringe was not cleaned, the initial glucose solution would be added up to the 1 cm cube or the 4 cm cube, which would alter the results. Okay, so explain why the clean syringe was used in step 3. So we say to avoid any contamination that would affect the final results. Coming to the next B part of the same question, a student extended the investigation by using more concentrations of glucose. Calculate the volume of 5% glucose solution and distilled water that are needed to make 5 cm cube of a 3% glucose solution. We are going to refer to step 2 and 3 where we got the hint for this question. Okay, so in step 2, it's mentioning that use a syringe to put 1 cm cube to labeled 1% and 4 cm cube to labeled 4%. Okay, total volume would be 5 cm cube. That is what we did by adding of water. So coming back to my calculation, 1 cm cube for 1% and 4 cm cube of water was added to make 5. So hence here if we have to make 3%, we add 3 cm cube of glucose and the remaining 2 cm cube of water to make it 5 cm cube. So when they ask you a question, volume of 5% glucose solution, your answer will be 3 cm cube. And the volume of distilled water, 3 plus 2 is 5. So we are adding a remaining 2 cm cube over here. Some people would also like to use the formula C1V1 is giving you the CFVF value. So what is this C1V1? I will try to explain that as well. So C1 will be the initial concentration, which will be 5%. Okay, and the volume is what we had to find here, the volume. Okay, then we are talking about the V1 of the volume to be found. So then we are talking about the CF is the final concentration, which is 3%, which they have asked you to make. Okay, here 3% of glucose solution, and Vf is the final volume, that is 5 cm cube. Okay, so we can substitute this in the formula as well. 5 into V1 is equal to 3 into 5 cm cube. If I put V1 here and I'm going to substitute again, so 3 into 5 upon 5, I can cut this 5 and 5 and your answer will be 3 cm cube. This is also how you could get the answer. Okay, but this is a rather lengthy method, so sometimes when we use logic it makes a lot of sense, because these papers will consume a lot of time for the graphs and the drawings of the diagram, etc. So you should know how to do the tricks. Moving on to the next question. Sports drinks often contain glucose, which is a reducing sugar. State how you could test the sample for sports drink for reducing sugar. So we will write the procedure. Take 2 cm cube of the sports drink. For reducing sugar, we use Benedict's reagent. So Benedict's reagent is taken. In the Benedict's reagent, heating is a very important process. So never forget to write this. Heat it in a water bath at 80 to 95 degrees Celsius for 2 to 3 minutes. Observe the color change from blue to brick red. So the original color in the test tube would be blue and the final color would be brick red. Okay, so just for your reference, this is what the color would look like in the initial blue, and the final brick red would be on this particular color. Okay, so this is our Benedict's reagent color change. Moving to the next part. Again, a very simple question asks that the ethanol emulsion test can be used to show that the sample of food contains fat. State the result for a positive test. So a milky white emulsion will be observed. If you remember the fat test, you are taking the ethanol solution and adding fat and ethanol, and you are going to stir the test tube. Stirring is a very important process over here, shaking or stirring. Now you add 2 cm cube of distilled water and a milky white emulsion will be observed. So that is the observation they are asking. Suggest one safety precaution that should be followed while using ethanol. We know that ethanol is flammable and that is why we have to keep it away from the flame. It's a 1 mark answer, but I'm giving you some more points. You can cover the container to prevent evaporation as well. And always use safety goggles while doing this kind of experiment where there are flammable or hot objects or hot solutions. Coming to the next part of the question. Plan an investigation to determine the effect of exercise intensity on the heart rate in humans. As we understand that as the exercise intensity increases, the body's demand for oxygen will increase and the body will start making more carbon dioxide. Breathing rate and heart rate are not the same. Heart rate is the number of beats and breathing rate is how fast your diaphragm and external intercostal muscles are contracting. So at one point of time, lactic acid will start to accumulate in the body. So if we see it with a graph, okay, I am going to draw that this is the level of glucose in the body and the red one I am putting it as the level of lactic acid in the body. As the level of glucose increases, oxygen increases during the exercise, sorry, my bad, this was oxygen. Okay, so as the level of oxygen increases, there will be one point where the body will not be able to take in any more oxygen. Okay, and at that point of time, lactic acid is also increasing, and at one point the level of lactic acid will increase, and this is called as the oxygen debt. After that, there is a recovery phase where the oxygen levels intake will be still higher only, and that is how the body will get rid of this debt. So we need to plan an investigation to determine the effect of exercise intensity on the heart rate in humans. So we start with the independent variable always. So I am designing this experiment on the treadmill. So the speed of the treadmill will be the independent variable. Dependent variable will be the pulse rate before and after the exercise. The procedure goes as that I have selected two individual males of same age and BMI. So these become my constant variables. I am talking about the same sex organism or same gender as we speak about. Okay. Then we talk about the same age and we are talking about the BMI. So these are my constant variables. Their pulse rate was recorded using a heart rate monitor or a smartwatch at the start and the end of the exercise. They were asked to first walk at 2 meters per second and then run at 8 meters per second on a treadmill for 5 minutes with a break of 20 minutes in between. That 20 minutes is important because we need to bring the heart rate back to normal pulse rate and again then start with the next exercise. The exercises were repeated 3 times to find the average. And during this exercise, now see again, this is a kind of an experiment like one of my earlier videos where there was no safety precaution actually like a lab coat, goggles, etc. So every time it need not be the same thing. So here I have mentioned that during the exercise there was a medical supervisor available to monitor and they were asked to wear comfortable footwear. You could also say that they could run on a flat surface, but I have already spoken about treadmill, so I didn't put that point. Okay, and you could also mention that the person could inform if they were feeling uncomfortable at any point of time before 5 minutes. Okay, try to stick to the dotted lines which are provided. Try not to exceed beyond this. I hope there are no doubts. If you have any doubts, please feel free to put it on the comment section. I will be more than happy to solve your doubts. Moving to the next part is your drawing question. So they have given you a necklace starfish and they have asked you to make a large drawing of the necklace starfish. So here I have drawn. Now how are they going to mark you for the same? So there has to be a single outline with no shading. So you cannot have any lines like this. It has to be one continuous line like this. So that is your one mark. Then another mark is the width of the drawing should be greater than 93 millimeters. So that is the magnification or how big the drawing is done. Again, as I've mentioned earlier, you are expected to use 70% of the place provided. Again, just for your explanation, I'm drawing these lines to show you that this is the space that you are supposed to occupy. That is, and you have to cover at least 70% of the place provided. Okay, so in this much place, you are supposed to draw this part. Okay, next labeling mark would be 5 arms and enclosed shape in middle that doesn't touch the edges, so that's the enclosed middle space, and the indication of where the color changes all on all limbs. Always remember, whatever is dark and visible except for the individual cells, you should draw it in the diagram. Okay, moving ahead. Measure the length of line PQ in figure 3.1. So this was the line that we were supposed to measure, which was approximately 94 millimeters for me. Um, they have asked you to find the actual diameter of the necklace starfish using the formula and give your answer in two significant figures. Please do not forget to read this. So substituting in the formula, 94 divided by 0.32 will give you 293.75. They've asked you two significant figures. They have already put the units as millimeters, so your answer is going to be 290. Always remember to do the calculations in millimeters. Next question reads, figure 3.2 is a photograph of a spiny starfish. Figure 3.3 is a photograph of a necklace starfish. The magnifications of the photographs are not the same. State two ways the spiny starfish in figure 3.8 differs from the necklace starfish, and one way it is similar. So it's a 3 marks question, 2 marks for the differences and 1 mark for the similarity. Now, they have told you to write state two ways the spiny fish differs from the necklace fish in figure 3.3. Okay, now they have specifically mentioned do not put any references of the size because it was said that the magnifications are not the same. So that is why we cannot say that is big or small. First difference that I would like to point out is that the spiny starfish, obviously, as the name suggests and the diagram shows, it is spiny like this. Okay. And the necklace starfish, it has a smooth edge or there are no spines. Here, I could say that it is light in the center and the necklace starfish is dark in the center. I could say that there are 5 ossicles. You can see this 5 things. 1, 2, 3, 4, 5. So there are 5 ossicles in the center. Here it does not have 5 ossicles in the center. Then I could say they are more uniform shaped limbs, and here we are saying that they are less uniform shaped limbs. It was very easy to see that these are quite straight limbs and these are like, showing these kind of curvy edges. Okay, so we could say straight limbs and bent or curved limbs. Similarities, when we go to, we can say that they both have 5 arms, which is your most obvious answer. They have circles in the center is also your answer. Okay, and they have radial symmetry could be one of your answer. So moving to the further question. Seven-armed starfish live in the sea. Scientists investigated the distribution of the seven-armed fish in one region. Photographs of 20-meter square area of the seabed were taken at different depth. The depth of the water in each area was measured when the water was at its highest level, that's during a high tide. The number of seven-armed fish in each photograph were counted. All photographs were taken in the same day in July, so that there is no seasonal variation, time of the day like hot water, cold water, etc. Nothing was going to change the number of the fishes that were found. So they had kept quite a bit of the constant variables or something that could not hamper or affect the final results. State two ways that the method used by the scientists ensured that the number of seven-armed starfish counted at different depths could be compared. So, we could say that, all that I have circled over here. So, it was taken in the same region. The same size, that is 20 meters square of the sea size was taken. It was done in the same day of July. Depth measured at the same time, that is during the high tide level. So all these were the levels, the constant things that they had kept for comparison. Now they showed the results and they are asking you to plot a histogram. Histogram is touching bar graphs, which is a way of counting continuous variables. Okay, so they have specifically asked you to plot a histogram. Do not forget to label the x-axis as the depth in meters. I have told you earlier in the videos that this is always your independent variable and this is always your dependent variable. Independent variable always goes on the x-axis and dependent variable always goes on the y-axis. So the histogram has been plotted using the scale. Again, the rule states that you have to use at least 70% of the place provided. So rationally look out for the bars and the scale that you are going to prefer. Okay, and do not forget to label in the x and the y axis. Always put these lines in the bars because sometimes if the pen is too light, I mean the pencil, if it's too light, it will not be visible in the paper. So it's better that we put these lines so that they are nice and clearly visible. State the depth range at which most seven-armed starfish were found. So if we go from the graph, we can see that the most were found between 10 to 20, and also from the numbers that we have to use from here. So it's 10 to 19.9. Since they have asked you a range, you have to write 10 to 19.9. Using the information, calculate the total number of seven-armed fish that were photographs at all depth. So all depth means we are going to add all of them and use the value to calculate the percentage of starfish that was photographed between 0.0 and 9.9. So 0.0 to 9.9, our number was 337, and we have to first add 337 plus 1486 plus 1243 plus 241 plus 121, which will give you a answer 3428. Now we have to find it out in the percentage, express the percentage as a whole number. So we have to multiply by 100. So 337 divided by 3428 into 100 will give you 9.83. So your answer is going to be 10% because they have told you whole number. 9.8 will be rounded up to 10. So this is the whole paper which has been discussed. If you have any doubts, please do get back to me in the comment section. Don't forget to like and subscribe so that you can stay updated for the further videos. All the best. Thank you.